So first, I'd like to thank Curtis and Joel for having invited me to present at this workshop, and to NIMBioS for organizing this. So basically, just a change of topics. Wanted to discuss informatic approaches to analyzing metagenomic data, and I'll look at a particular flavor of it today, and that's what we'll go over. But basically, we've seen from Jill's talk and others' talk yesterday, just a flavor of how sort of metagenomic analysis is done. You have a collection of metagenome from a collection of organisms, do your sequencing. You have this notion of wanting to bin and assemble organisms so that you can then get at their metabolic and functional composition in order to study various aspects of the community. What I'm going to focus on today are a couple of topics related to sequence assembly and searching, homology detection, but basically in the context of proteins. So I'm going to talk about assembling protein sequences from metagenomic data, and how to do homology detection using assembly as a step. So again, microbial communities, I think we're all familiar with this. Just to set the stage, often collection of organisms, taxonomically distinct, different abundance levels, and their genomic compositions can be, again, depending on the available carbon and energy sources and the complexity community, their compositions can be quite different. And of course, one of the challenges in metagenomic analysis has always been this question of, can you infer complete or near complete genomes of species from the sequence samples? Again, they're downstream that has a lot of utilities. But of course, we and others found, especially analyzing medium and more sort of complex communities, that this has often been a challenge. In fact, Jill's first project, the acid mine drainage study, really set a very high bar in terms of this question of, can you reconstruct genomes? For the global ocean sampling project, one of our first questions was, could we tease out genomes of the abundant organisms from those samples? And try as we might, we just couldn't get at it. It was a hard problem. And now we know some of the reasons why, right? Strain variations, genome rearrangements, all kinds of, and sequencing depth, obviously, another big component. It has been a challenge. And same with some of the other, couple of other larger projects, the MetaHit Consortium project, looking at the human intestinal tract communities. Essentially, this notion of fragmented assemblies with short contigs and a large fraction of the input reads being unassembled. Well, as we know, assemblies serve as substrate for a whole bunch of downstream analysis. And there are consequences of poor assemblies, namely, you get fragmentary gene sequences. Annotation can be a problem. And essentially, annotation analysis is restricted often to assemble parts of the communities, which is not a good thing to do. On the other hand, when we talk about sequencing technologies, you look at Illumina's 2500 machine. It can spit out in about 6 days or so nearly a tera base pair worth of data. That's about 6 billion base pair paired and reads. About 125 base pairs long. And really, if you're taking this approach of trying to annotate every last read, if you will, that can be computationally quite prohibitive. So our focus really was this question, that is, well, okay, if assembly is a bit of a challenge, why don't we, and our interest is in reconstructing protein sequences, why don't we jump straight to that question? Can we, given a set of reads from a metagenomic sample generated from a community, can we reconstruct complete or near complete protein sequences from it directly? And why would something like this work? Well, prokaryotic and viral genomes have high coding density. What that means is that majority of the reads will contain portion of a gene. There are good de novo gene finders for metagenomic data out there. So a couple of examples, FragGeneScan, Metagene Annotator. So what that means is you could use these guys to, one of your favorite gene finders to predict fragmentary protein sequences. I'm referring to those as short peptides here. You could do this quickly. And The third reason why this might work is the whole notion of redundancy in the genetic code and the amino acid conservation, the fact that it extends over a larger taxonomic range compared to nucleotide conservation. So the SNPs, the polymorphisms that are a big challenge for nucleotide assemblies may not be as much of a challenge when you're assembling in the amino acid space. So these were sort of our three pieces of sort of rationale for wanting to proceed with this. And the approach that we took is again parallel to the nucleotide assembly or the assembly of genomes with short-read technologies, essentially this notion of a De Bruijn graph or a Kamer graph. So just to present, just defining what it is, given a sequence, a Kamer graph basically stores information about the Kamers that are present in the graph. And an edge basically represents a Kamer that's found in the sequence. And the nodes, K+1 mer, and the nodes represent Kamers. And two nodes are connected. If the prefix, if this is the K+1 mer, and that's the prefix of the K+1 mer, and that's the suffix of the K+1 mer. De Bruijn graphs have been used quite a bit, again, as I said, for nucleotide assemblies. And in fact, many years ago, it was used by Edure and Waterman to do assemblies from sequencing by hybridization techniques, and then followed by various different flavors of assemblers that have come out. De Bruijn graphs offer a nice compact representation of your data. And for our framework, really, we construct De Bruijn graphs using the amino acid alphabet. Okay, and essentially, we end up using a k value of 6. So basically, we construct De Bruijn graphs of with Kamer 6. Our framework really is quite simple. So we start with the metagenomic reads. We use a gene finder to identify these short peptides. We construct a De Bruijn graph with these short peptides. And then we do an informed traversal of the De Bruijn graph using things like depth of coverage information, meaning how many times you've seen a particular Kamer in the graph. And you do that, and we use that to sort of output paths, useful paths in the De Bruijn graph. And then we have a post-processing steps that sort of cleans up. So the whole idea is that the way we do this traversal is how we're trying to get at what we think are true protein coding sequences. And just to give you a flavor of what this is, I mean, suppose this is your graph, and this is sort of the path that you've traversed thus far, essentially the way we look for the next jump, if you will, is this notion of what's called a thickest extension. That is, you're here, you essentially look for overlap, reads that overlap with this particular node, right? And that have at least an L-back, or what's called an L-back overlap. That is, there are reads that can be placed with an overlap of L, at least L here. Okay, and we have a few little steps that take care of things like repeats, and we have a sort of procedures for repeat handling and some rules for when to stop your extension. So once we get this initial set of paths, then we go through a few series of steps to kind of clean up your initial paths. And we also have a post-processing step to sort of handle overprediction by gene finders. We have this implemented, and then we compared this to the alternate strategy, which is a strategy, I guess, that we often use, which is given a set of reads, we would do an assembly based on using one of these nucleotide assemblers, and then we could call proteins off of that, and we run with that, right? So we wanted to compare that strategy with the strategy that we take. That is, the output from our short peptide assembly framework, the proteins that are output, how do those compare with respect to the proteins output using one of these assemblers? Again, just setting the stage, evaluation, I mean, we use these different measures, specificity, sensitivity, which is defined really at sort of this, using this notion of a length match. So if P is a set of all the amino acid sequences or the paths that are output by a particular method, either by SPA or by the nucleotide assembly-based method, and let R be your reference protein sequence, then you can define specificity and sensitivity using this notion of a length match. Okay? And it's not very different from the standard definition, except that we have this length match component in here. So we had several different data sets that we evaluated our approach for. These couple of first two data sets are pretty easy examples where, if you just take fragments from genomes, are you able to reconstruct them back? And I'm not going to present the results here, but sure enough, we do really well here. But here's where a bit more of the interesting component comes into picture. And so I guess one thing we haven't touched upon in this meeting, but I think it's worth noting is, as we develop methods, appropriate data sets for evaluating methods is often a challenge. And so simulators, right? Simulation, we often tend to do it using simulated data sets because you can then fix sort of the ground truth and try to compare things based on that. Simulators in metagenomic space, I know there are a couple of ones out there. We actually use this thing called, this simulator called Metasim out of Daniel Houston's group. It's a pretty good simulator. But I think that there is quite a bit of open room to play in that area as well to generate the right kind of, the simulators that can model the microbial community the right way. That is strain variations, genomic rearrangements, taking into account evolution as well. This is a pretty simple model where what we did was, we started with 25 genomes, in this case from the oral microbiome community. And then we let them evolve using a Duke-Scanther model of evolution using Metasim. And then out of that, we generated about 500 genomes, did a shotgun sort of sampling of them, if you will, and generated 100 base pair reads. And that's sort of what went as input to each of these methods. Okay, so basically we had about 115 million reads as input. And the reference protein set from this entire collection of genomes was identified by taking the proteins from these genomes and then clustering them at 95% identity. The overall performance looks for SPA is actually quite good compared to the other approaches. And just to show you, and by the way, this is done at 0% sequence error. In the publication, we actually report using error profiles as well, so up to 1% sequence error, how the different methods perform too, we've looked at. But just to give you a sense, I mean, the sensitivity of SPA is better than that of the other approaches. And again, this is where the length component comes in. So that if you're asking, this question of, well, in terms of recovery, that accounts for 90% of the sequence length coverage. But this is we get about 80% sensitivity or so compared to the other methods. And you can see how that value actually goes up if you decrease your length constraints. Specificity is lower than sensitivity, but it's still sort of higher than the specificity of the other methods on this data set. And we did the same for another data set, in this case from the marine environment, some of your favorite candidates. Data generated the same way. And we get a similar sort of set of results as well in year 2, where the performance of SPA using frag gene scan as a gene finder tends to be quite good. With respect to read assembly, so the other big question we also ask is, what fraction of your reads get into assemblies? And of course, the other big concern about this sort of a recovery process is, what fraction of your predictions could be chimetic? And by definition of chimera here, I mean, we take a prediction, we ask if there are portions, non-overlapping portions of the predicted protein sequence that have matches to 2 different parents, if you will, at a really good identity, so 90+ identity. And so that's our definition of a chimera. And we find that the chimetic rates in all of these methods are actually quite low, including SPA. And the percent of reads that are in assemblies are SPA is among the topmost compared to the other approaches. Okay, so I think it is a sort of a promising approach using simulated data sets. When you look at real data, where of course you don't know the ground truth, so we took 2 HMP samples, one with 40 million reads and the other with 86 million reads. So we couldn't compute, of course, sensitivity here, but we can look at specificity if we use something like NR maybe as your reference set. Okay, so We took the predictions from each of these methods and used and asked, okay, do you detect these do you find them in NR, and use that sort of as your ground truth in some sense. Specificity values are not as high as we saw in the simulated data set, but it's still sort of for SPA compared to the other methods is much better. And I think the reason, of course, we suspect here is that coverage. If you guys remember, for the simulated data set, we ended up using something like 10x coverage of the genomes. In real data sets, often with these community, with the amount of data that's generated, the coverages of individual strains of genomes are much lower. I would say probably on the order of 2x or 3x. Of course, for the abundant ones, it's much higher. But by and large, for a good fraction, it would be much lower. And that's what we suspect that it's the specificities are lower here. So just as a summary, this framework offers you the possibility to bin the reads into protein groups, if you will. Okay, and again, we're doing this at the amino acid level. So what's the utility, you might ask? Well, you can use this for all kinds of downstream analysis, right? You could then get back into your original nucleotide space if you wanted to do something akin to like haplotype phasing, if you will. Meaning if you wanted to get back at strain reconstructions of proteins, you could then use your nucleotide information and do that, right? So there are lots of sort of downstream analysis, and of course, then you can use taxonomic. You can use those protein bins to do taxonomic profiling of the communities as well. Okay, so one of the challenges with SPA initially was that its time complexity, and the fact that when we were doing this sort of LBAC overlap kind of thing, we were essentially using a bunch of set operations. We thought a little bit through about that and realized that, in fact, all of these steps could, in fact, be modeled using, or the queries could be done really efficiently using a pretty popular data structure that's used in the computer science community, suffix arrays, right? And it's also used quite a bit in genomics as well. So suffix arrays are linear space and linear time query data structures that allow you to capture read overlap information and do efficient querying. And using that, just the path identification step in itself, using a suffix array-based setup compared to the original one gives you a much better speed up. So you get about 12-fold, 13-fold improvements on the large data sets here. This is with the 100 million reads. And of course, if you look at the overall running time, you get about close to about 5.5 times speed up. So it makes it a bit more feasible to apply it on data sets with 100+ million reads. So that was the first piece, which is de novo assembly of amino acid sequences with the goal to do reconstructions of full-length and nearly full-length protein sequences. The second problem that I wanted to briefly touch upon is homology detection. So the framework is this. Suppose you're given a query sequence, protein sequence, and you have a database of protein sequences. Standard thing. You want to identify sequences in the database that are homologous to Q. A popular solution, of course, you could use something like BLAST, right? But what happens when the database contains short peptide sequences? Say we've run FragGeneScan or Metagene annotator on the individual reads, and that's your collection of database sequences. Well, what you will find is that the performance of BLAST, in fact, would be dependent on the length of the sequences in the database, okay, but also the degree of conservation of the protein family. And if you're lucky, and if that fragment really hits a conserved part of a protein of the query sequence, you will find it. If not, it's going to be a challenge. You will not detect it. The big question is, can we do better? And this is where the idea that these fragments are coming from the same community comes into use here, right? So what if the sequences are from the same protein family, as is in the case of a metagenomic data set, where you're doing random samplings of your genomes. And therefore, what you will have is, again, it's a function of the sequencing depth, obviously. But given all the other reasons, as I mentioned earlier, about conservation of redundancy in the genetic code and so on, we will get, we have a much better chance where short peptides from the same family you find them to be overlapping, right? So if they are overlapping, well, what if we, as we're doing homology detection, what if we did assembly at the same time? Okay, well then you can improve your chances of detecting homologs. And that's really the approach behind this next framework I'm going to talk about. But just to show you an example, what we did was we took a couple of seed sequences from a PFAM family. I think one of these Lig-T phosphoesterases. We took this query sequence. We took another member of the same family, chopped it up into 30 amino acid long sequences, and just used BLAST to see if we can, how many of these sequences we can reconstruct. Now, if you take the full-length sequence, this is sort of the alignment and the e-value of that alignment, which is obviously pretty good. But the fragments, you see that their fragmentary, the alignment scores for the fragmentary sequences are quite low. But if we did an assembly of these overlapping fragments, and then the score kind of jumps. And this is sort of the idea behind our next approach, which is called GRASP, or Guided Reference-Based Assembly of Short Peptides. Again, as a framework, we could think about your original de Bruijn graph formulation, right, with the short peptides. Given a query sequence, what you're trying to do is you're trying to identify paths in the de Bruijn graph that have good homology with good alignment scores, if you will, with this query sequence. But we need to start somewhere, right? And for example, in BLAST, what is done? Well, BLAST takes your sort of standard sort of seed and extend approach. The idea being you look for these short words that are shared between the query and the database sequence, use that as an anchor, and then you extend out till you can't anymore, and then you output what are called these high-scoring pairs, or HSPs, right? We use the same approach here. But again, the challenge is where do we start? Because think about the number of candidates that one would have to look at, right? Even if you're because it's a trade-off between the number of false extensions you end up doing versus the number of candidates versus the number of good extensions, right? And one idea that's been used previously, and which is what we used here as well, is look, you start with your original amino acid alphabet. You can, of course, do what's called you can reduce, into a smaller amino acid alphabet based on the similar sort of physical and chemical properties of the amino acids. And this sort of an approach, using reduced amino acid alphabets, have been used quite a bit for speeding up searches, homology detection searches. So RapSearch is one such program that is supposed to be a faster version of BLAST, and uses precisely this approach. So in that paper, actually, this figure is from that paper, where they actually looked at, for different alphabets, different reduced alphabets, they looked at what are called these coverage and efficiency scores, and sort of use that to kind of pick, which of these alphabets to go with. And we ended up playing with 2 of these alphabets, both of size 10. And so essentially the idea is this. You start with a query sequence. You look at it in reduced amino acid alphabet space. You take your database sequences, convert them into reduced amino acid alphabet space as well. Start with exact KMR matches, okay, in the reduced space. And then you start doing your alignment extensions and alignments in the original space, okay? So the idea here is you have your query sequence. Let's assume that's your region that's been aligned. And then you're essentially looking to find overlaps, meaning overlaps of or overlapping short peptide sequences in the database with that particular sequence. And again, that we can do this using this notion of a data structure of suffix arrays, and we kind of extend this. And in each step, essentially, what we're doing is, as we're doing these extensions, essentially what we're doing is what's known as a banded alignment of the query sequence region with the sequence that's being considered. Okay, and essentially this is used to identify overlapping peptides, or contigs, if you will, that have good scores when aligned with the query sequence. And of course, we can, we have this notion of a certificate where we can actually convey the evidence that a given short peptide is a homolog by virtue of the fact that it overlaps with a series of other short peptides whose total alignment scores are good, meet the cutoff. So we took this approach, idea, and evaluated it against a few other methods. So your standard method of BLAST, where, remember, the query searched independently against each short peptide, and SciBLAST, where, as you guys know, the iterative version of BLAST. FASTM is another sequence search program. And we used 4 different data sets. In this one, the idea was we took sequences from unrelated PFAMs. So we had 4 unrelated PFAM models. We took seed sequences from those models, randomly chopped them up, and used the query from one of them to ask, are you detecting the homolog? So we pooled all of them together, right? And here we find that if you look at sort of the ROC curve, we find that the GRASP approach gives you really good results compared to the other approaches. We also did the same thing, but this time with PFAMs from the same clan to make. So this is a slightly harder problem, right? So, and in this case, we actually looked at the glycosyl hydrolase superfamily and took about 4 to 5 different PFAMs, took the seed sequences from there, chopped them up. And the same thing, we took queries from one of the families and asked, if you did your homology detection using GRASP or BLAST or SciBLAST, what are the homologs you're identifying? Okay, and so obviously the performance is not as good as when you have unrelated PFAMs, but it's still better than the other approaches. Then we looked at a couple of other sort of metagenomic data sets. So the idea here was, you have a metagenomic data set. In this case, we took the simulated marine data set that I mentioned earlier, and took glycolysis genes from an organism that's not in the data set, and took their protein sequences as queries and asked, can you detect their homologs in the short peptide database that you constructed from the simulated marine data set. And again, you find that the performance of GRASP is sort of better than that of the other methods. In this last data set, what we did was we looked at the same thing, but with only respect to the marker genes. So these are the marker genes that are highly conserved. As you know, so this is your standard set of, 40-odd genes that are used for taxonomic profiling. There you find that, the performance of these methods come very close to each other. In fact, SciBLAST does a slightly better job compared to GRASP in that case. And again, I think that has to do with the fact that these are highly conserved proteins. So there's not much difference in the performance of these different methods. Okay. We also looked at a human saliva data set. And so here the question was, again, so we have a real data set. We've taken, and we took, again, glycolysis pathway sequences from 2 organisms. One, which is Streptococcus, I think it was abundant in this sample, and another from Lactobacillus, which is in very low abundance. And again, the question was, using those as hooks, meaning those as query sequences, can we identify homologs in the data set? How many can we identify, right? And here you find that, again, it's the same question because we don't, same problem with the earlier situation where we don't know what the sensitivity looks like because there's no ground truth there because it's a real data set there. So we can address specificity and we can also address a number of hits that are identified. And essentially the take-home message here is that specificity levels are pretty good for all these different methods. In terms of the number of identified reads, so the number of homologs that are detected, GRASP actually tends to outperform the other methods. In fact, compared to BLAST, I think it identifies 3 to 5 times more the number of homologs. And so, again, I think from a homology detection perspective, this is a potentially sort of promising, useful method. Again, one of the challenges here has been speed. So the computational efficiency of GRASP versus something like BLAST or SciBLAST. Keep in mind that those programs do only remote homology detection, whereas I guess here we're doing assembly as well. So so what that means there is that GRASP is definitely going to be slower than the other methods. But we have, in fact, just recently sped it up by about 30-fold. That allows us to now kind of do queries against databases consisting of about 12 to 15 million short peptides in the order of about a few minutes or so. So that's sort of bringing it down to the level of Cyblast, if you will, in terms of speed. So again, I think as a paradigm, this notion of assembling an amino acid space and then using it for either for de novo assembly or for homology detection, I think it's a worthwhile idea to pursue. And I wanted to acknowledge my postdoc and staff scientists in my group, and also the NSF for funding. So, thank you. I'll take questions.
So for metatranscriptomics applications, what's the pre-processing cleanup that you'd recommend before feeding sequences to SPA?
Right. So metatranscriptomics, of course, get rid of all the, even if you do depletion, RNA depletion and stuff, first thing is to get rid of all the rRNA sequence and stuff. So I guess using RiboPicker or any of the other favorite tools out there. And then it really boils down to identifying the short peptides, right? Meaning winnowing down to what we think are protein coding genes, right? So whatever steps are required to get to that point, so removing rRNAs and non-coding RNAs are really the main steps. And then applying your favorite gene finder. We found that, especially for Illumina data, and I guess, if you're not too concerned about, if your technology that you're using is not too prone to sort of frame shift errors kind of thing, then you're fine with going with something like, metagene annotator, something that does not have to do sort of frame shift modeling, as well. So you do the cleanup, get these short peptides, and then you should be able to let go with SPA.
I think you did your development with 100 base pair reads.
Yes, I mean, so in fact, we can apply to 100.
I was going to ask, how does the situation change when you've got 250 base pair reads, and then what happens when you go to PacBio? Have you looked at trying to apply it to that technology?
So with questions, things like PacBio, as you get longer, the homology detection problem, of course, becomes easier. I mean, in terms of the alignment pieces and all that. But the way I've been thinking about with newer technologies and the use of something like this is, I think, again, as we were discussing yesterday, this notion that cost-effective nature of things as well, right? So I would suspect that we would be using this when we have a combination maybe of long PacBio reads plus short reads, Illuminas. So when we talk about, the idea there is that we can use maybe the genes that are predicted from PacBio or molecular or whatever it is as hooks into identifying additional homologs from your metagenomic data that you might have sampled at a graded or sequenced at a graded depth using some other technology. But with respect to your other question, is what happens to the ability to reconstruct, the problem becomes easier in some sense, right? But the remote homology problem, so at that point you might say if you have 250 or even PacBio long reads, at that point you don't need GRASP. I think you don't need to assemble is what I'm saying. But that other situation, that is, you have a long enough protein you've identified, maybe from an abundant organism in your community, and then trying to get all the other homologs of lower abundant organisms for which you only have sort of short reads. That you can do much better. Okay. So.
Great talk.
Yeah.
Also, you mentioned molecular, which was great. So I wonder about memory requirements and scalability of your assembler in general compared to the other, do you need a big box to run your...
Yeah, no, thanks for that question, yeah. So the main, so let's see, going back to SPA, I said we were talking about, the graph, the de Bruijn graph sizes that we're dealing with, right? We're using K of 6. So we're talking about 64 million, in terms of just starting the sizes there. But really the main thing comes in as you're capturing the input sizes using the suffix arrays. And what we found is that, for the large data sets, about 110 million reads, we can, it takes about 80 gigs or so. So what we're talking about is about, I think with the 128 gig machine, you should be able to process data sets, at least for SPA, you should be able to process data sets on the order of 100, 150 million reads or so. To do
what about the really big ones? Like what about the really big ones, like the 2-4 billion type of data sets?
Well, yeah. So there we can adopt more of a hierarchical approach as well if you needed to, right? So we could do this thing where we say, well, look, if we have the 6 billion size data sets, we could partition it randomly into chunks of 100 million kind of stuff, do SPA on each of these individually, and then start to kind of group things together. The idea being that each of these data sets, okay, if it gives you back partial or nearly full-length protein sequences, then we can start to bring in other kind of clustering tools as well, right? So CD-hit and other kinds of things that we can use to cluster things to. So that's another approach, but we haven't applied SPA to billion-size data sets yet. Yeah. But it's a good question.
So I thought I'd give voice to a conversation we were having on Twitter about how your gene finders work on short reads like this. So they found a paper that said that 0 stop codons is the requirement, but as far as I understand, gene finders often presume a start codon and not too many stop codons. They're about the right size. There's a lot of things that don't apply to short reads.
So and that's the I would say kind of a remarkable thing that all these constraints, yet they've tended to do really well, right?
So you just throw it in there and press go?
Well, no. So basically, in terms of some of so if you look at the sort of the ideas behind some of these concepts, right? So metagene annotator, for example. Let's take a step back. Prokaryotic gene finding, obviously, it's much simpler compared to eukaryotic gene finding. Prokaryotes, you're essentially trying to figure out the right reading frame, right? And of course, there's a challenge of figuring out whether the right start site ought to be, right? So metagene annotator, for example, looks at dicodon frequencies and sort of takes, given a read, computes a GC composition. From that, it tries to extrapolate and figure out what the codon pattern ought to be and uses that to figure out which frame. FragGeneScan uses hidden Markov models to be able to figure out the frame, and if there is a frame shift, to be able to do the jumping of the frame as well. These methods are really good. I mean, I would say if you look at the performance on data sets, you get, I think, 95% plus accuracy for these methods. And there are other ones as well. I mean, I think Orphelia, a whole bunch of other different methods as well. So you might think it's a harder problem as you get and yes, and there are some limitations, obviously. I mean, in the sense, I think even some of these things don't even identify things shorter than 20 amino acids and stuff, right? So there are those constraints, for sure. But 100 is not a problem whatsoever, yeah.
Yeah, so this has come up a couple of times. So for my lab, we do sort of longer 250, 300 base pair reads, right? And then we're directly doing functional profiling with, say, Diamond or something against the KIG database. And it seems like so now we have sort of the direct approach, we have DNA assembly approach, and then we have protein assembly approach. And there's obviously disadvantages and advantages in different scenarios, but I'm wondering sort of what that, if anyone's looked to see what that length cutoff is, where we should switch from one to the other, the speed trade-offs, the effect that gene callers might miss genes. Right. Has anyone sort of done that sort of cross-sectional study that I've just missed? Or
so when we were doing the GRASP work, we actually did look at the effect of, I don't have the results here to show you guys, but we did look at some of the effect of the read length. So all the way from 100 to 250 bases, what happens, right? And again, as I was mentioning earlier in reference to Jill's question, at some point, meaning I would say 250 to 300 amino acids. So let's take a step back. Average bacterial gene size, right? 300 amino acids, right? So the moment you sort of come down to being able to get, half of that or even a third of that, you can get some nice hooks into your setup, right? So I think the moment you start getting into 300 base pair long reads, high quality reads, I mean, then you might be in good shape to be able to identify homologs without the need for this sort of an assembly kind of approach, where you might say, well, I'm just going to do sort of your standard master-slave sort of alignment, if you will, and then try to assemble it or analyze it post afterwards, right? But the other question, what are the kinds of things you can do down the line? I would say, if we're using this sort of an approach where you're identifying homologs or binning amino acids, using the amino acid space to do assembly. But down the line, I mean, the next thing you can do if you want to tease out individual organisms would be, let's revert back to the nucleotide space. Let's revert back to the raw reads that were used in here, because in that you can use to tease out information about maybe even strain level. We've not done that yet. I mean, we've been playing with some of the data, but we don't have any proper results on that yet. But that's an approach we want to take as well. So maybe you can use that to estimate genome abundances and stuff too.